Hey there, I am about ready to do another installment on my series of amino acids and proteins. And the topic for today's video is to talk about the acidic and basic properties of amino acids. So I will make my face a little bit smaller and get it out of the way. So as a reminder, an amino acid group contains both a carboxylic acid group and an amino group. So now I'm going to make my face bigger. So I'm going to bring this model up. So here is my central carbon, my amino group, my carboxylic acid group, the R side chain, and the hydrogen. So this carboxylic acid group and the amino group show acidic and basic properties respectively. Now I do have this in the zwitterion form. You'll notice that my carboxyl group is showing a COO, and it has a negative charge, so that is the carboxylate ion there. And then rather than NH2, in this zwitterion form, I have the NH3, which will have a positive charge. So I thought that visual might help a little bit better than just reading the words off of the screen. So when we're talking about that zwitterion, the H, which is very loosely held on this carboxylic acid group or carboxyl group, those are two different ways that that group can be used interchangeably, either a carboxyl group or a carboxylic acid group. This loosely held hydrogen does get transferred to the amine group here, and we end up with an NH3. So this produces dipolar, meaning in this molecule there are two regions of charge, one on the carboxylate side with the negative and on the amino side with the positive. But the whole thing is electrically neutral. So here is an amino acid that has a full COOH carboxylate group, and here is the basic group H2N, and here is the zwitterion form. Now, how amino acids exist in the body is very dependent upon if it is in an acidic or a basic medium. So if it's traveling through fluids in your body at different pHs, it's going to impact what this looks like. So sometimes if you are in a certain pH, you might pick up an extra hydrogen, and for example, if it's an acidic medium, you'll pick up an extra hydrogen and it'll attach itself here, leaving just this behind. Or if you're in a basic environment with hydroxide OH group, you might take the hydrogen from this protonated amine group here. So the next couple of slides are going to go through those scenarios. So if you got jumbled up in the words I was just saying, which can happen sometimes, I know that when you listen to things, I'll be going through some examples shortly. So I have the two models next to each other, the amino group with just the NH2 and the COOH. And with this one, I'm going to activate the pen here for a bit. With this one, this has lost its hydrogen, so this region of the molecule is going to have a slight negative charge, and that hydrogen came over here. So this is protonated now, and this takes on a plus charge. But the thing still overall has as many negatives as there are positives, so it is overall neutral. So if we take an amino acid in its zwitterion form and put it into an acidic environment, it is going to pick up, remember, in that acidic environment, we have lots of protons or hydrogen ions floating around. That's what makes an environment, that is one of the classifications of an acidic environment, is hydrogen ions in solution are greater than hydroxide ions. So anyway, this will get picked up and it'll get reattached right here. And we end up with this structure that takes on an overall positive charge. So we still have our protonated amino group over here, and now we have the hydrogen. So this leaves an overall positive amino acid. Now the same amino acid could then go ahead and travel to different parts of the body where it encounters a more basic media, higher pH. And what can happen there is, in a more basic media, our hydroxide ions are more concentrated compared to the hydrogen ions, and the hydrogen from that protonated amine can be pulled off of that, leaving behind that NH2 group again with no charge, and we get the production of water. So if we have a zwitterion form amino acid and we put it into a base, it ends up taking on an overall negative charge because we still have this carboxylate end here, that COO with a negative. So this is just a summary of the two things that can happen, and also a representation of how amino acids can act as buffers in the body to help maintain stable pH levels. So one thing that can happen is if amino acids are present and we're in an acidic environment, in order for it to help prevent it from becoming too acidic, this hydrogen ion can be picked up and keep a balance on that hydrogen ion concentration in solution if things are getting out of whack. On the other hand, if the pH is getting too high and we're ending up with more hydroxide ions, amino acids can act as a buffer to pull hydrogens off and remove the hydroxide from solution so that the pH doesn't get excessively high. So in addition to playing a role in protein synthesis in the body, amino acids also do act as buffers. So to summarize, the overall charge on an amino acid molecule is going to depend on, number one, what it is, how many regions of charge there might be on it to begin with, and number two, on what the pH is of the environment that it's in. So every single amino acid has a pH at which the net positive and the net negative charges are evenly balanced, and that is called its isoelectric point. It's not always neutral. Each acid has a unique isoelectric point, and here is an example. So for basic amino acids, that isoelectric point ranges from 7.8 to 10.8 on the pH scale, and at that point, at whatever that pH is for that specific amino acid, the net electronic charge will be neutral. So for neutral, that isoelectric point ranges from 4.8 to 6.3 on the pH scale, and for acidic, it is lower, between 2.8 and 3.3. So the overall takeaway of this is every single amino acid has a pH in which the the amount of negative charge is equivalent to the amount of positive charge. There's no net electronic charge, and that is called the isoelectric point. So if we look at, just changing gears a little bit here, with these acidic and basic properties occurring in amino acids, we have our acidic end and our basic end. When an acid reacts with a base in a neutralization reaction, one of the products is a salt and one of the products is water. So amino acids can take on the characteristics of salts. For example, if you isolate an amino acid, it is crystalline. An example of this is something that I purchase from the over-the-counter is an amino acid called L-lysine, and L-lysine helps me when I start getting cold sores. I read about it on the internet. I actually looked up some scientific peer-edited reviews of the ability of L-lysine to help with skin conditions and the assistance of clearing up cold sores and even preventing them. It does show to be effective for some people, so I started taking it regularly, and it has minimized the number of cold sores that I get. If I start getting a cold sore because I haven't been taking my L-lysine, then as soon as I can just take a bunch of them over a series of days, and it clears it up quite a bit faster. So a reminder that the reason that we get that left-handed amino acid, is because that is what our bodies use for the production of proteins in protein synthesis. When those amino acids are coming together, it has to be the left-handed variety. So speaking of amino acids in our diet, there are some essential amino acids, and the characteristic of the essential amino acid is that our bodies don't make them. Our bodies cannot synthesize them. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 essential amino acids. Some sources you see will say 9 essential amino acids, so I think that varies depending on who you talk to. But since our bodies can't make it, it needs to come from dietary protein. So dietary protein can be categorized as either a complete or incomplete protein based on whether it has these amino acids. So a complete protein supplies all of the essential amino acids, and sources of complete proteins include meats, dairy, and things like that, eggs, poultry. And an incomplete protein is deficient in one or more of the essential amino acids. So there's this notion out there that people who take on a vegetarian or a vegan diet are not going to get enough protein, and they most certainly can. Oh, I didn't realize there was another thing here. It says that corn does not supply lysine. A person following a vegan or vegetarian diet, as long as over the course of time you're eating foods that contain the essential amino acids, not necessarily in a single food item that you're eating, but over time, if you are getting that intake, you'll be just fine. So here are some examples. Grains, nuts, and seeds are high in methionine and low in lysine. Legumes are high in lysine, but low in methionine. Corn is high in methionine, but low in lysine. I am not sure what. Oh, there's a little joke. And a woman dressed up as corn. Isn't that fun? I got this slideshow from another instructor at some point in time. So complementary proteins are things like beans and rice. If you eat rice and beans together, you will get all of the essential amino acids. Peanut butter on bread, beans and corn, black-eyed peas and cornbread, nuts and soybeans. So sometimes if you're preparing dishes, you can be mindful of getting the essential amino acids in one meal. However, again, it's not necessary if you are getting an intake, a regular intake of the essential amino acids in your diet over time. It is just fine. So if you haven't, if you're one of my students and you haven't been able to interact with this model kit in class, or you haven't seen this video, this is a video that talks about what determines how the overall shape of a protein, and it's a really good segue into the next topic, which is protein structure. So it provides some introductory concepts, and it's also very beneficial to watch if after we end up learning about protein structure, because it provides a really good summary and a really good review of the topics talked about with that. So the next topic in this series is going to be protein structure. And again, this is a really good video to watch before we talk about that, and it's an excellent video to watch after we talk about that. One problem with the video, I will forewarn you a little bit, is that some of it does get cut off, so you have to just kind of work through. There's a part in which I'm referring to text, and you'll just have to listen to what I say at that point, but it becomes pretty obvious what I'm saying. So thanks for watching my segment with proteins on the acidic and basic properties of proteins, and I'll see you next time. Thanks.